What's up? It's Ruan.
It's Bea.
It's Gail.
It's Rachel.
It's June.
And we are here to discuss
Cellular Transport!
Let's get started!
Have you ever wondered how you can smell perfume or how raisins are dried up? All of these things require the movement of substances by either passive, active, or bulk transport. In this video, we are going to discover how cells take in useful substances and remove waste using these three methods of transport.
Let's start with passive transport.
So what is passive transport? Passive transport is the movement of ions and other atomic or molecular substances across cell membranes without the need of energy input. Under this is diffusion, osmosis, and facilitated diffusion.
Let's start off with diffusion.
The smell of Bea's fart spread around the room by diffusion. Diffusion is the process in which particles spread out from each other. They move from high concentration to an area of low concentration. In diffusion, particles move down from the concentration gradient until they are evenly distributed. For particles to move like this, they need to be gas or particles of a dissolved substance. Diffusion allows substances to pass into or out of cells across the cell membrane, but they must be dissolved and there must be a concentration gradient that they can move from high concentration to low concentration. Gas exchange in the lungs. So oxygen moves from the alveoli into the blood and carbon dioxide from the blood into the alveoli are both great examples of diffusion. Carbon dioxide moving from the air into the leaves of plants also moves by diffusion, but it doesn't have to be a gas. Our digested foods like amino acids move from the small intestine into the blood also moves by diffusion. You can also have facilitated diffusion, where the movement is still down the concentration gradient, but special carrier proteins are required to enable the movement. Glucose and ions need carrier proteins to move across the membrane.
Osmosis is very similar to diffusion, but just for water, it is the movement of water into or out of the cell. Again, it is the movement from dilute solution, so high concentration of water molecules, down the concentration gradient to a more concentrated solution, so low concentration of water molecules. The water molecules move across a partially permeable membrane. Water molecules move at random, so some do go back across the membrane, but the overall net effect is that there is movement to the more concentrated side, or the side of less water, until equilibrium is reached, or until there is no net movement of water. Osmosis out of a cell can cause big changes. Animal cells shrivel up, and in plant cells, the membrane and cytoplasm shrink away from the cell wall, causing the cell to become flaccid. And if osmosis causes lots of movement of water into the cell, plant cells swell and become turgid. They have a strong cell wall, which prevents them from bursting, whereas animal cells don't have a cell wall, so they burst. The absorption of water by plant roots and the absorption of water in the small intestine and the colon all use osmosis.
We have three solutions. The first one is isotonic, the second one is hypertonic, and the third one is hypotonic. In an isotonic solution, the solute concentration inside the cell is equal to the solution outside the cell. Therefore, the amount of water transported into the cell is equal to the amount of water transported out from the cell. There is no net movement. On the other hand, in a hypertonic solution, solute concentration inside the cell is lower. Therefore, water is transported out from the cell. causing the cell to shrink in a process called crenation. Lastly, in a hypotonic solution, the solute concentration inside the cell is higher. Therefore, water is transported into the cell. The cells inflate and eventually burst in a process called lysis.
Now let's go to active transport.
In the first part of this video, we looked at diffusion for the movement of gases and osmosis for the movement of water. They were things that could easily move from a high concentration to a low concentration. So what is active transport for? Well, the clue is in the name, active. Work has to be done, so there is a need for energy. Active transport moves molecules from a low concentration to a high concentration against the concentration gradient. This is the opposite of diffusion and osmosis. Active transport is carried out by protein carriers within the cell membrane, and they have a specific binding site for exact molecules they are transporting. The substance binds to the protein carrier on the low concentration side and using energy from respiration. The protein carrier releases the substance on the other side of the cell membrane, where it is already at high concentration. See the difference between diffusion and active transport? In diffusion, no energy is needed because the molecules can just slide down the concentration gradient, where in active transport, they are joining the crowd and so more effort is needed, hence the ATP or energy.
Active transport allows cells to absorb substances from very dilute solutions, such as root hair cells taking in mineral ions on the soil, and in kidneys as we can hang on to the substances our body needs, such as glucose and sodium ions. Cells which carry out a lot of active transport often have lots of mitochondria to give them the energy they need. So from these two parts, you should know how cells take in substances and get rid of waste by diffusion, osmosis, and active transport. Just remember that for diffusion, the substance moves down the concentration gradient from a high concentration to a low concentration. Just think of a slide. You don't need any energy to go from high to low. It's easy, whereas active transport is the opposite. The substance needs to move up the concentration gradient from a low concentration to a high concentration. Think of climbing a ladder. You need energy to go from low to high up.
Lastly, let's discuss bulk transport.
The third and last type of transport is bulk transport. It is used for molecules that are too large to be moved by transport proteins. Vesicles take them into or out of the cell. During this process, the plasma membrane surrounds and engulfs the particle. This is known as endocytosis. Cells use three basic types of endocytosis depending on the size and nature of the material to be digested. Number 1 is phagocytosis, number 2, pinocytosis, and number 3, receptor-mediated endocytosis. If the material taken in is large, such as bacteria or a food particle, the process is called phagocytosis. Pinocytosis occurs when vesicles form around the liquid, or very small particles. During receptor-mediated endocytosis, molecules bind to specific receptor proteins embedded in a coated pit within the plasma membrane. When enough molecules accumulate in the coated pit, the pit deepens, seals, and is incorporated into the cell as a vesicle. The other type of bulk transport is exocytosis, which is the opposite of endocytosis. During exocytosis, membrane-bound vesicles move to the surface of the plasma membrane, fuse with the membrane, and then release their contents outside of the cell.
Thanks for watching! Hope you guys had a wonderful day. Bye!